Okay, students, we're going to practice writing lipid structures today. Okay, so this particular chart was in the PowerPoint regarding types of lipids. It's also in a handout that's called Writing Lipids. So it's in two different spots on your Canvas. And so let's take a look at how this will work. I'm going to add in, I'm going to make sure I'm on my pen. And let's go with blue, I guess. Okay, so these are our fatty acids, and if you notice, they're condensed structures. And the important part is that they have a carboxylic acid group here. One of our more famous ones that we'll use is palmitic and stearic and oleic for today's discussion. Okay, so what we're going to see, which we went through on the PowerPoint on types of lipids, is triacylglycerols are the type of fats that are stored in adipose tissues. And later in the semester, we'll talk about how they're broken down for energy. But for right now, let's just focus on their structure. So let me write down the structure of glycerol. So that's CH2OH. Okay, so there's the glycerol backbone. And what we're going to see is that this carboxylic part of the fatty acid is going to combine with the OH part to form an ester linkage. Okay, so there's a couple things that I think are really important for students. One is to understand that when they combine, they're going to give off water. And that the other thing to understand is that we're going to have to do some flipping around of these structures in space. So I'm going to rewrite palmitic acid, for example, as H, kind of the other direction. Do you see that? Okay, so you see how that is palmitic, just flipped around in space. So let's do another color. We'll have stearic acid. Okay, so there's stearic acid. So I just got it from here. And then our last one that we're going to add in is oleic acid. That one's this one. I'll use a different color for that guy. Guess we'll go with green. Okay, that one is an unsaturated fatty acid. Okay, so there's the three fatty acids. And now we're going to combine them to form a triacylglycerol. And what you want to think about is when they combine, and I'm going to circle it with another color. Let's see, I guess we'll go with, I'm not sure which one's going to be a good choice. How about this yellowish color? Okay, so what's going to happen is when they combine, that water's going to come out. So you could picture that you have this OH and this H are removed out. This OH and this H are removed out, that OH and this one H. Now, what's going to help us a lot is to take a look at kind of rewriting this part of the structure. So I'm going to go back. And I think what I'll do is I'll erase this part and kind of write it a little bit differently. Let's write it as its carboxylic structure, right? The COOH. So if I write it like that, the structure is going to make a lot more sense. So I'm going to rewrite the red one. Oops. Okay, and then I'll rewrite the green one. Oops. Okay, so now when we're going to go to combine them and remove that water, we're going to pull it all together. So it's going to be, I guess I'll use the color coding still. So it has that glycerol backbone. Okay, so there's our glycerol backbone. And then we will add in our fatty acids that have now formed an ester linkage. And what we're really drawing is a structure that is extremely hydrophobic. So this is going to exclude water. So when it gets stored in our fat cells or adipose cells, water is not going to be attracted to it. So it really improves the storage capacity for these fats. The other thing the reason I'm having you learn how to draw these is because later, when we start processing fats and we learn about how we process them for energy, one of the important parts about it is that it's very rich in carbon. So this is our storage molecules, adipose cells, fat cells, and they have a high potential for energy. But the other thing that you should look at, they're very rich in carbon, but they're also very large, bulky structures. So these don't dissolve in the bloodstream. So there's all sorts of difficulties and pathways that are needed to break them up and also to deliver them through the bloodstream. So they need special carriers. They're stored in fat cells, but they're not broken down. So they have to be transported. So the fact that this is extremely water insoluble, the structure definitely affects its function. And it means that it's harder for us to process fats for energy. But we'll come back to that when we do fat metabolism. Okay, so let's go to our next structures. So our next ones are the phospholipids. And the phospholipids are cell membrane lipids. And my main goal as we learn how to draw these is to really, when we go to the cell membrane lecture, that we have a really good understanding of how their structure affects their function and how it affects the way that the cell membrane is composed, like how it is in structure. So if you notice here, I'm going to grab the highlighter. Here's our glycerol backbone still. So that is still in the yellow. That is our glycerol. Okay, and I'll highlight that in yellow. So that's our glycerol. We still have 2 fatty acids. So you have fatty acid 1. Oh, shoot. Sorry, guys. When I switch too many colors and I have too much going on. So fatty acid 1 and fatty acid number 2. Okay, now here's the important piece. The glycerol backbone, what I want you to notice is that instead of putting the O on this side, okay, it's been flipped to the other side, and there's going to be a reason for that. And I'll tell you about that in a second. So it just got flipped. There's rotation in that bond. Okay, and then instead of just having a third fatty acid, what it's going to have is a phosphate group. And then that phosphate group, in the most simple structure, it could just have an H. Okay, but there can also be other things attached onto phosphate. You could have serine, you could have choline, ethanolamine, inositol. I won't stress this last one. So this, I'm going to put an X. You don't need to know that guy too much. Okay, so when we're looking at these, what you'll notice is once they attach to that phosphate group, you have a really, really charged polar group on the end. So this piece here that I'm circling, that's our polar head. And then this part back here are the nonpolar tails. Okay, so let's practice drawing one. We're going to do a phospholipid, phosphatidylcholine with palmitic acid and oleic acid. Okay, so we're going to start with our glycerol backbone. Oh, let's do that spinning thing on the end there. Spin it around to the other side. Oops. Okay, if you do that, it's just going to make it a lot easier to understand the cell membrane structure. Okay, so what are we going to do? We're going to take oleic acid. And for this, oh, I should have mentioned, you'll have those structures, so you're not memorizing these. So that's good news. I should have said that earlier. Okay, so I'm going to go with palmitic. Okay, so there's palmitic. Okay, the other one's oleic. I'll do a different color for that. And then where it says phosphatidyl, okay, that means we're going to have our phosphate group attached. And then we're going to have choline. So let's do choline. I can put these guys in different colors if we want. Let me go back. Let's see, I guess I'll do red. So here's phosphate. And then for choline, we can do another color. Guess we'll do blue. So for choline, I'll write it over here. Now you're going to have that on your sheet. All of these are going to be given. So what you have to do is just combine them. I'm going to condense choline structure from what's on that other sheet. Okay, so these would all be given you have to know how to put them together. Okay, so let's start. Let's remember that we are going to have the water come off of here, right? We're going to have water come off. We're going to have water come off of there. Okay, and then let's get going and writing that part. I'll start over here kind of in the center. Okay, so there's our glycerol backbone. And then now I'm going to combine it with the palmitic acid. Okay, now I'm going to add on attach the oleic acid. Okay, so that's not really it's no different than what we just practiced with the triacylglycerol. Okay, so there's your 2 fatty acids. And then now what's going to happen is that you're going to attach the phosphate group onto here. So I'm going to highlight it. One of these oxygens has to go. So you're going to see what it would have been is, again, a water coming off of there. So we can get rid of that OH. Okay. And the other thing that I realize I'm missing here is, so let me erase that real quick. That would have 2 hydrogens on there. Okay, so with this in mind then, I'm going to do something a little bit different with that green. That's going to be attached from that O. Okay, so let's go ahead and write that in, the phosphate group. Okay, so that phosphate group comes in there and attaches. And then you have essentially what's a phosphoester bond. So the regular esters with the carbon group. So this is an ester bond. This is a phosphoester bond. Okay, now in the most simple phospholipid, there would be an H here. But we are now going to attach the choline from this OH up here that I've done with the arrow. So once again, you want to picture that you have water coming off. Whoops, I didn't mean to erase that. I meant to highlight it. So that's going to have an OH group there. And you can picture an H is going to come off of here. It's also, there's H plus ions. So that's going to come off. And so now we will attach the choline onto the end of that. So let's go ahead and do that. I'm going to erase that H. And then I'm going to attach the rest of it, which is too thick there, huh? I'm not sure why that's highlighted so thickly. That looks very strange, so I'm going to try to erase that. Try to get back a more normal bond there. So that OH on the choline is removed. Okay, and now hopefully you can see that we have a really, really polar group. Polar charged group on one end, and then you have a hydrophobic part here. Now, this is a really important slide because later, when we do cell membranes, they're going to abbreviate this like this. They're going to show it like that, and they're going to have it be like this. And what I want you to understand is that obviously that's a very big simplification, that it's not a blob with two tails, that you actually have charged polar groups on one side and fatty acid chains on the other. So what I'd like you to do later as we go to cell membrane is to understand that this is an abbreviation of this big giant structure. Okay, so we have one more structure. And here's the sphingolipids. Sphingolipids dominate the nervous system. And in some ways, they're a little bit, in my opinion, they look scarier, but they are a little easier because the structure, so this is sphingosine is the backbone. It's not glycerol. So you don't have a glycerol backbone, you have a sphingosine, which has this part is part of the structure. So that's always there. That's always the same. And you'll only have one fatty acid attached. And then just like we saw with the phospholipid, see here this OH, okay, that's our more simple one, but you could get rid of that H. Oops. Let me go back, sorry. You can get rid of that H. I don't know why it did that to me. Let me go eraser. Oh, there we go. Okay, good. Oh, I don't know why my highlighter went a different color. Oh, sorry, guys. Okay, I'll stop messing with that. So you can get rid of this H and then attach any of those other groups: ethanolamine, choline, serine. Okay, so all right, so let's try to draw these real quick. So we're going to have a sphingolipid, a ceramide with palmitic acid. So from our previous discussion in the lecture part of this, the ceramide is our most simple one. It means that this R group here is an H. So that is our ceramide, that's just a hydrogen. Let's see if I can get that to look a little bit better. So our ceramides just have a hydrogen on them. And then this one says palmitic acid, so that's the fatty acid, right? The one fatty acid. So this is a little bit, in a way, easier, even though I know it looks more intimidating. Okay, so let's go ahead and draw it. We'll draw the entire sphingosine as written. That one's always there. That's part of the backbone. And then your fatty acid, palmitic acid, I'll use a different color for that. So palmitic you're going to have that available to you. Okay, didn't draw that super well. Let's do it like that. Just makes it easier to see. Okay, so there's our palmitic acid. And now we're going to attach it. But remember that water's going to come off. And so what's going to happen there is I'm going to highlight that. Well, maybe not. I'm struggling with that highlighter right now. Oh, there we go. This OH is going to come off as a water. Okay, so what's going to happen now is I'm going to attach it. Go back to the pen. Purple. It's going to look like this. So you have to get comfortable with kind of flipping them around. So this one here is attached. And in this diagram, that's that carbon there. Okay, and then I'll finish off the structure. And I am going to keep the polar head on this side. Okay. All right. The last one, phosphosphingolipid. If you can, I would pause right now and see if you can pull it all together. Reminder, you're going to have the structures, you're just putting them together. And so let's take a look. Pause right now, see if you can do it. And then you can press play again when you're ready. Okay, so I'm going to keep going then. I'm going to have my same exact backbone. Okay, so there's the same backbone, but now my fatty acid is oleic acid. So it's going to look like this. Okay, so the fatty acid is, again, you're going to get rid of that OH group that it has on the end to attach, and you form an amide linkage here. Okay, and then the last one is, now this says it's a phosphosphingolipid. So what we're going to need is a phosphate group. And then I need choline. So the choline would be the CH2, CH2, and CH3, 3. You'd have that structure. Okay, now when you look up choline, remember it's going to have the OH group on the end. Oops. I was going to abbreviate it a little bit more, but let me just keep it consistent with how I've just drawn it. Okay, see this OH group? That's going to go out as water. So this is not shown over here. Oh, except, I'm sorry, I forgot the O on the phosphate. Should have kept that on there, I'm sorry. Ah, boy. And I'm not going to redo this, I'm sorry. Sorry, to be patient with me. Too many things going on with the pen and the colors and all that stuff, so. Gave it a whirl. There we go. Sorry, I meant to have that on there. So, kind of to recap that piece with, there has to be oxygen linkages here, but there won't be two oxygens. So like here, this OH will go, but the O on the phosphate would stay if you want to think of it that way. This O stays, but this oxygen that it would have attached to doesn't, because water comes out. And if you notice, this has a really large polar part to it. Okay, so then when we see these guys, it'll look like that. That's going to be their abbreviation. Two nonpolar tails and a very charged polar head. So there's a lot to practice on the homework, and let me know if you have questions. We can go through this again in an office hour Zoom if you'd like, when I see you next week.